Hi everyone, I'm really glad to be speaking with you today, and what I'll be doing right now, or in the next couple of minutes, is I'll be giving you what the OFIS program calls the lab tour. And I suppose the purpose of this is to expose you to a quote-unquote real-world lab, and show you what a real-world lab is like, and maybe a little bit of how it operates. And you could be doing all sorts of different things in the lab. You could be studying cells, that's sort of what I do. You could be studying entire organisms. You could be outside of the controlled environment of a lab, such as outside in a rainforest, let's say, or somewhere else, collecting insects or looking at trees. And labs are just all different. And so, I guess just to also provide you some context then, I'll be talking a little bit about my research first. So, what I do is I study something called graft versus host disease. And it's sort of a weird thing. It's a potential complication of something called a bone marrow transplant. Bone marrow transplants are really just a fancy term, if you're not familiar with it, for saying that you're going to give someone immune stem cells. And immune stem cells are also called hematopoietic stem cells. And so you might hear someone call a bone marrow transplant a hematopoietic stem cell transplant. And that's abbreviated as HSCT. They mean the same thing. And really, what happens when you give someone immune stem cells is you're giving them the precursor cell to all blood cells. Immune stem cells are the precursor to red blood cells, white blood cells, platelets, all the blood cells in your body. And there are multiple reasons that you might want to do that. One of the biggest reasons, by far, is if someone has a bone marrow cancer or a blood cancer. And we would hear of that as leukemias, lymphomas, or multiple myeloma. Another reason that we might want to give someone new immune stem cells is if they have some sort of a genetic condition, like SCID, which is severe combined immunodeficiency. You might have heard of someone, or you might have heard of Bubble Boy, and Bubble Boy is a person who had this condition, and he actually got a bone marrow transplant, but unfortunately he had an illness a couple months after the bone marrow transplant. It was later found that his transplant was actually contaminated with Epstein-Barr virus, which caused a lymphoma that ultimately killed the boy, which is unfortunate. But that sort of exposes you to the fact that there are actually two different types of bone marrow transplants. There is a type of bone marrow transplant where you can give someone immune stem cells, hematopoietic stem cells, from the own person. So you can say that I'm going to give Bob a hematopoietic stem cell transplant, a bone marrow transplant, and I'm going to collect immune stem cells from Bob and then give them back to Bob after I give Bob, let's say, a high dose of radiation or a high dose of chemotherapy to kill Bob's pre-existing immune stem cells, which literally creates space within the bone marrow, physical space within the bone marrow, for the new immune stem cells to successfully take up residence. And the term for that is called engraftment. So you can give Bob cells that you already collected from Bob beforehand, or you can give Bob cells that you collect from a different person, like Alice, let's say. And that could be a good thing. But there's also a potential complication when you give Bob immune stem cells from someone else, because these immune stem cells will eventually give rise to all immune cells in someone's body. And if you give them immune cells from someone else who's not identical to them, who's not their identical twin, those immune cells are going to have a different genetic makeup than the person who's getting the transplant. So Alice's cells are going to be genetically different from Bob's cells, and they might recognize Bob's cells as foreign and react to them and attack them. And you might wonder then, why even give Bob someone else's cells? It turns out that there is a potential protective effect of giving a patient someone else's immune cells. So if you have some sort of a blood cancer, let's say, or a bone marrow cancer again, like a leukemia, it turns out that with current advances, these immune cells from Alice, which might recognize Bob's cells as foreign, will also be much, much better at recognizing Bob's cancerous blood cells as foreign and react to the cancer and attack the cancer and destroy the cancer. And that's called a graft versus tumor effect. And that's something that can be very protective. So it turns out actually that if you give Bob Bob's own cells versus if you give Bob Alice's cells, in the case of certain cancers, such as multiple myeloma as a specific example, it's actually more beneficial to give someone cells from a donor, from a different person, and not from themselves. So that's sort of the setup to what I study. And it turns out, depending on the exact paper in the literature that you look at, that graft versus host disease unfortunately occurs in between about a third and half of the patients who receive bone marrow transplants. And so really, it's a big problem. What I do mainly in my research is I do single-cell RNA sequencing. I look at the genes being expressed in immune cells of patients who receive bone marrow transplants and try to compare them. Ask ourselves if patients who do go on to develop graft versus host disease have something different about their immune cells compared to patients who remain free of graft versus host disease. And this might help us, this might inform us about, let's say, a potential pathway that's involved in graft versus host disease that we could target with a drug or a medication. And actually, this is not totally related to my research, but the data set, the blood samples that I'm analyzing were collected as part of a study that has been finished that looked at whether a particular drug called abatacept was effective in preventing the acute form of graft versus host disease. And it was in fact successful at doing that. And so abatacept has now been FDA approved for the prevention of acute graft versus host disease. But that still leaves the chronic form of graft versus host disease, for which there is no current FDA approved treatment. And so yeah, so now let's go ahead and take a look at our lab. Just generally, every lab would have some sort of a bay like this where people can work. And in this building I'm in, there's a whole bunch of these bays. So if I just walk around, you can see that there's a bunch of rows of these lab bays, and behind me as well. And we can go ahead and take a look at what's in one of these bays. Well, actually, let's first talk about just some general lab stuff. So I would say that in any sort of lab, it's very common to have some sort of personal protective equipment. And that would commonly include a lab coat. So I'm going to go ahead and put on a lab coat. And gloves. So I'm also going to go ahead and put on gloves. Depending on exactly what you're doing, you can also have protective safety goggles, which would sort of look like that. I'm going to go ahead and put these down because we won't be needing them today. So just taking a look at this lab bay here, some of the really common items used in labs are pipettes like these that you see over here. Pipettes are basically devices that let you measure a very precise volume, often a very small volume, and dispense them. And this pipette is what's called this pipette dispenses up to 2 microliters of fluid, which is a really small number. If you think of things in terms of a centimeter or a millimeter is 10 to the minus third of a meter. A microliter is 10 to the minus sixth. So that's 1,000 times smaller than a milliliter. Pipettes, how you use pipettes is you take it, and I'm sure that all of you have done this in some sort of a high school bio class. You put it into a tip. Okay, you can't see that right now. Here's a different one. Take a pipette. This is a P1000. It dispenses up to 1,000 microliters, which is also the same as a milliliter. Here's a box of pipette tips. You push it in, like that. And to get some sort of liquid, you push the pipette down a little bit, down to what's called the first stop. You insert it into your liquid that you get, and you lift it up slowly. And I'm just going to get rid of this tip. So there are red bins around in the lab, red bins that are used for just disposing of biohazard. And that's where we get rid of most trash that we work with. There's this device here that looks sort of odd. it's connected to a vacuum, and it sucks air basically. If we're doing some sort of a DNA extraction protocol, for example, oftentimes we'll add a lot of reagents that either help get rid of proteins that might chew up the DNA, and we want to suck out that solution. And we're going to use a device like this, for example. We can also see a bunch of equipment here. This is a machine used for something called single cell RNA sequencing. This black machine here, and what it does is it adds a unique DNA barcode to a single cell at a time, literally a single cell. And then when you use a different machine to sequence that cell, all of its transcripts, all of its RNA will have been tagged with that unique DNA barcode. And so you can know exactly which genes are being expressed in a cell and at which levels. And that's really relevant to my research because that's basically what I look at. There's a machine here called a thermal cycler. And what people use this machine here for is cycle through a bunch of different temperatures. And that's something that's very useful for a procedure called PCR, polymerase chain reaction, where you want to amplify a piece of DNA. So you cycle through different temperatures that either dissociates the DNA, anneals the DNA with certain enzymes, and then a high temperature where the enzyme is going to work to elongate a piece of DNA. There's also a centrifuge here, which is very, very classic lab equipment, and centrifuges are used for spinning samples. So if you're going to process blood, for example, you want to separate the different fractions of your blood. You have some serum, you have a thin layer of what we call peripheral blood mononuclear cells in the center, and then you have a pellet at the very bottom. Here we have a machine that is called a tape station, and what it's used for is it's used for analyzing the quality of DNA that we might want to extract. This is a machine called a Kia cube, and it's used also to extract DNA. We can take a look at one of our rooms that we use to work with cells. And so this is a cell culture room that you can probably find in a lot of labs across the country. And since I didn't actually introduce myself, my name is Edward, and I was a former immunology master's student at Harvard, and right now I'm a research tech working in the same lab that I got my master's in. And you can see that you have an incubator here, and incubators are important because cells are sort of difficult to work with sometimes. In order to make a cell happy, you need to give them a very precise temperature. You can see that the incubator is set to a temperature of 37 degrees Celsius, and they also want a very precise CO2 level. You can see that that's 5%. And they are also very picky about the humidity, and you can see that that's set to 88.1%. What we have here are what we call biosafety cabinets, and they're important to use when we're working with anything that's potentially pathogenic, let's say, or potentially dangerous, because they keep dangerous things within the hood at all times. There's a purple glow in this cabinet, and that's because we're opening a UV light right now, which is supposed to sanitize it and disinfect everything. You can see some pipettes up here, so really common laboratory instruments. Within a biosafety cabinet, you can see that, well, there's a device that's just like a pipette. It's like an automated pipette, basically, because it quickly draws up liquid without you having to continually press or depress a button. We have some more pipette tips on that end there, and we have some containers in there to hold tubes and various stuff. We have a refrigerator here. We have some pretty common reagents in the refrigerator. We have this bottle here it's labeled RPMI. RPMI is a very common medium that we use to grow cells. So for example, we have a project in the lab where we're trying to grow CAR T cells. Those are T cells that are modified to, let's say, better fight cancer. And so that's what we use culture medium for. We also have, in this freezer on top here, we have FBS, that's short for fetal bovine serum. That's also a very common cell culture medium. Basically, it provides nutrients for cells to survive and grow. Some more gloves. This device, a cellometer, is used to count the number of cells in a particular sample. So if you were to, let's say, isolate immune cells, isolate those peripheral blood mononuclear cells from a blood sample. You would want to count to see how good your sample was, to see whether you got a high or a low amount of cells. There's a microscope here, and I can turn it on and turn it off. Here, this is another centrifuge. It's used to spin samples. It separates things based on their weight. A much larger centrifuge here. And I believe that's basically it in terms of the lab. More interesting stuff. Just, we have a bunch of conical tubes. Very standard stuff you see in a lab. You have a pipette here. Actually, one important thing about labs is that you want to have, about working with these well safety cabinets, is you want to spray everything that you put in it with ethanol before you put it inside. So let me just open up this fume hood. And there's a line that tells you, there's a line that very precisely tells you how high you can lift up the glass. And what you want to do before you put any object in the fume hood, including your hands, is to spray it with ethanol. And ideally you should do it outside, but I'm only using one hand here because I'm holding I'm holding the computer with my other hand. So you spray something with ethanol. And this is 70% ethanol. You sort of wipe it and you put it in the cabinet. I'm going to I also sprayed this container of PBS, which is phosphate buffered saline. It's a common medium. I'm just going to open it. I'm going to keep the lid on because I don't want to contaminate anything. What you can do with this, it's got a pipette A label there, is you can open up your pipette tip, insert it into the pipette, and then insert it into the container, press a button to draw up the solution. You can see that solution is coming up here, and then press another button to withdraw the solution, instead of the solution going down again. And then when you cap the container, you put the pipette tip back into its packaging, and then dispose of the pipette tip in one of those ubiquitous red biohazard containers. So yes, I believe that's it for the demonstration today. Thanks for watching along.